Okay, so let's continue on with our discussion of cells. So we know now that cells need to be small to increase their surface area to volume ratio so that their transfer of materials into and out of the cell are more efficient. And we had talked about how there are two main kinds of cells, prokaryotic cells and eukaryotic cells, and talked about the bacteria, and that there are certain things that all cells have in common, which were these basic features. But now we're going to look at more specifically eukaryotic cells. So it says here a panoramic view of the eukaryotic cell. A eukaryotic cell has internal membranes that partition the cell into organelles. So you have these membrane-bound organelles that are separate from the rest of the cell. And this is really important because these organelles are specialized to do particular functions. And so therefore, it congregates all the materials that are needed for that function in one spot. So it's way more efficient that way. So like for instance, the mitochondria is one organelle, and this is where cellular respiration takes place. So there are enzymes to speed up these chemical reactions, and all the things that are needed for cellular respiration are in this mitochondria instead of just free floating anywhere inside the cell, because then it would be harder for those molecules to come into contact with each other and those reactions to occur, and so therefore it makes it way more efficient this way by having these specialized kind of rooms kind of inside of this cell. So internal membranes facilitate cellular processes by minimizing competing interactions and by increasing surface area. We're going to see different organelles have ways to increase the surface area, in which therefore increases the reactions and things like that can occur in the organelle, like a mitochondria. So membranes and membrane-bound organelles in eukaryotic cells localize or compartmentalize cellular processes and specific enzymatic reactions. Enzymatic reactions means they use enzymes to speed them up. So that's what I was talking about, a process like cellular respiration in the mitochondria with the enzymes and with all the things that are needed for those reactions. So the basic fabric of biological membranes is a double layer of phospholipids. And so we learned about this in chapter 5 with phospholipids. Phospholipids make up the membrane, and so of the cell, but also of the organelles within the cell. So therefore, the membranes within a cell, even around the organelles or around the cell itself, have that in common, in that they are a double membrane of phospholipids. So remember that the polar heads of the phospholipids face towards the water. The hydrophobic tails are on the interior because these are hydrocarbons and want nothing to do with the water. All right, so plant and animal cells have most of the same organelles. So this right here, is it a plant or an animal cell? Are you able to tell? This right here is an animal cell. And it says here, right here, figure 6.8a, so that refers to your textbook. And so it shows you all of the different organelles within it and what they look like and so on. And so we're going to talk about most of these as we go along throughout the chapter. I do want to point out that animal cells are one major type of eukaryotic cell. The other major type of eukaryotic cell is what is it? A plant cell. And so this is a plant cell here, and so this is 6.8b, so I'm going to label this a plant cell, and it also has various organelles and so on as well. And you'll notice that a lot of the organelles are the same. So between the plant and the animal cell, a lot of them are the same. And so let's write here, down here, the organelles that are in animal cells but not plant cells. Now remember, all cells have those varying characteristics that we looked at in the first page. So all cells have the plasma membrane, they have the cytosol, they have a chromosome, and they have ribosomes. So that's going to be true about bacteria cells, the prokaryotic cells, animals, and plants, the two types of eukaryotic cells. So let's look at these. What are in animal cells but not in plant cells? So when we look at here, things like the endoplasmic reticulum, there are the endoplasmic reticulum in both of them. So you can see ER and smooth and rough ER. ER stands for endoplasmic reticulum. So the things that are on this picture that are not on the other picture, I'm going to put little bullet points here, are lysosomes. And we'll talk about what those are. We're just talking about the differences between them now. Centrosomes with centrioles, and we'll talk about what those are as well. And then flagella. But I'm going to put here that they are present. There's an exception in plant cells, but are present in some plant sperm. So there's some plants that actually the sperm swims to the egg in the fertilization process. So that's the exception there. All right, so then let's look at the opposite here and look at in plant cells, what types of things are in plant cells but not animal cells. So, first one is site of photosynthesis. Chloroplasts. Second thing, this big thing right here, in plant cells it takes up the majority of room in the cell. It's called the central vacuole. Around it, it has a tough cell wall. Why is it tough? What molecule is it made out of? Did you say cellulose? Because that's, remember, one of our polysaccharides that we learned about. And they have these things called plasmodesmata, which are kind of like tunnels between the cells. And we'll talk about those in a little bit too. All right, so there are some differences that are unique to plant and animal cells, and then there are some things that are found in all eukaryotic cells, like the smooth and rough ER, the Golgi apparatus, the nucleus, and so on and so forth. So let's take a look at all these different types of organelles one by one, the organelles that you need to know for this class and be familiar with. So the first one is the nucleus. So the eukaryotic cell's genetic instructions are housed in the nucleus and carried out by the ribosomes. So the nucleus contains most of the cell's, what is it, DNA. All right, most of the DNA in a eukaryotic cell, because remember in a prokaryotic cell, there's no nucleus, so therefore the DNA is not contained in those. And then the ribosomes use the information from the DNA, so the DNA contains instructions to make what? Instructions to make proteins. And so the DNA contains that instructions. Those instructions are copied on a molecule of mRNA and goes to the ribosomes to make proteins. All right, so that's the big picture there. So let's look at the nucleus and the structure of it. The nucleus contains most of the cell's genes, because these are, remember, part of DNA, and is usually the most conspicuous organelle, meaning that you are able to see it. Usually when you look underneath the light microscope at a cell, you see the nucleus. And so and inside of it, what you don't see is the DNA. And so the DNA, remember, one DNA molecule has many, many genes, can have hundreds of genes. So gene here, a gene here. So genes are segments of your DNA. And those genes code for making proteins. And so there's instructions here to make proteins. And that's what gives you your trait. The nuclear envelope encloses the nucleus, separating it from the cytoplasm. So that's how it's compartmentalized, is it has this envelope, they call it, and And we're going to see it's a double membrane. So the nuclear envelope is actually a double membrane, each membrane consisting of a lipid bilayer. So this picture here is showing you, so this is a transmission electron microscope picture of the nucleus. And so what they did is they enlarged it and made a computer-generated picture of it. And they cut out a cross-section. And we're going to see here, so this is the nucleus right here, the whole thing. This is so it's a sphere. And they chopped it out like this, all right? And so we're going to draw some things in there and look at it. So let's look first at the cross-section here of the membrane. So we're going to look right here. All right, and if we enlarge this part here, let's see what we get. All right, so what you end up having is a double membrane. And so right here, and it goes like this right here. So therefore, what that means is that there are two membranes around the whole nucleus. So that's a double membrane. That's what that means. And this is I'm going to put a picture here, a colored picture, so that you can see here. So you can see here, see how it's double membrane? All right, so there's one membrane, two membranes. All right, and then you have these little pores through here, and the pores are like little like poking holes in the membrane. So they're little holes that allow things to go into and out of the nucleus. So right here is the pore right here, and so I'm putting these in pink. So this allows you to see the hole in between there, so it allows things in and out. That's your pore complex there. This is what it would look like from the outside. We're looking at the cross section here, and so that allows things to go into and out of the nucleus, that pore. And so when I say instructions are copied onto mRNA from the DNA, how does it get out of the nucleus? It would go through that pore. All right, and so you can see here on the outside of the nucleus, this is the surface of the nucleus. These are all those pore complexes. So these are all little holes in the nuclear envelope. Okay, and so that's what that is here. This little picture here is an enlargement. And to relate this up to the phospholipid bilayer, there are two bilayers here, all right? So therefore, there are two layers of phospholipids, hence the double membrane. So that's the first part. That's what separates the nucleus from the cytoplasm. So that's what we just talked about here. So let's look at what else is inside of the nucleus. And so let's see here. I'll use orange. There's a dark spot right here. It's kind of hard to see. It's the nucleolus, all right, in orange there. So what in the world does the nucleolus do? So let's write over here. So a couple things. So it's in the nucleus. It is where our RNA is made. So our RNA is ribosomal RNA. Our RNA, or ribosomal RNA, is made here. All right, so that's going to be used for our to make RNA, and then the rRNA is used to make ribosomes. And once the ribosomes are made, the RNA mixes with proteins and makes the ribosomes. We'll learn later that ribosomes are made up of a small and large subunit, but that's where they're made. And then the ribosomes actually lead through the pores and go out into the cytoplasm. So that's the purpose of the nucleolus. And so underneath the microscope, when you look at a cell, you can see the nucleus, and sometimes you can see that dark spot inside of that, and that's inside of the nucleus, and that is the nucleolus. And then also, I'll pick green here, there's something called chromatin. So chromatin, usually you can't see underneath the microscope, and I'm just going to make it kind of stringy like this. All right, inside of the nucleus. So let's talk about what chromatin is. So I'm color coding here. So if we look here, I go down here. This is chromatin here. It's trying to show you here. I want to explain what it's trying to show you. What chromatin is it's DNA wrapped around proteins called histones. Okay, so what this is we have, like, this is your DNA in green, and your histones let's make in red. So I'm going to make histones red. Here's my histone proteins. And they have these little, like, that's why it looks like these things coming out here. They have these little things that come out. That's what they really look like. But and then the green part is the DNA. And so the DNA wraps around these histone proteins. And the whole thing, so this is DNA, these are the histones, and the whole thing collectively is called chromatin. All right, so it kind of, the purpose of the histone proteins, I like to use the analogy of a spool and thread. So like if you think about sewing thread, if I just had a ball of sewing thread or a string of sewing thread, and it was really, really long, and I bunched it all up to fit it into a small space, let's say like a small little container, it would maybe knot up and so on. But if I take it and I wrap it around a spool, I can take that long thread, still put it in this tiny area, but it won't knot up and it'll keep it organized. And so that's the same thing with DNA. So the DNA in our cells is very, very long, a whole chain of nucleotides. And to keep it organized, we wrap it around these histone proteins, and then they kind of bunch together, and that's what your chromatin is. And so that's what we're seeing here. So this is, chromatin is found in a nucleus, found in a non-dividing cell. So if a cell is not getting ready to go go through cell division, you would find it in this loosely packed DNA around these histone proteins and in this chromatin form. That differs, we'll learn a little bit later, that differs, we're going to talk about it in the bottom of this page here, from chromosomes. You probably have heard of chromosomes. So what happens is this chromatin, when a cell is getting ready to divide, all of these histone proteins come together and tightly packed together into this short, stocky little structure. So now all the histones are all together, all right, and so you still have your DNA wrapped around your DNA, and so on. But you get this shorter, thicker, stockier structure because it's all condensed together. And that is what's called a chromosome. All right, in chromosomes, this only happens where it bunches together and forms the stockier structure. This only it happens when the cell is dividing. So this is found in a dividing cell, when it's getting ready to go through mitosis or meiosis. You probably remember those terms from biology. And so that's what happens. And so that's the relationship between these two. And chromosomes often, sometimes you can see them underneath the microscope because when they're shorter and stockier, they're easier to see. When they're loosely packed, like in the chromatin, it's thinner and harder to see. All right, so that's the difference between those two. So let's look at underneath this picture here. So what regulates the entry and exit of molecules from the nucleus? What were those? They were the pores, right? The pores regulate the entry and exit of the molecules. And in the nucleus, DNA is organized into discrete units called, all right, and I'm going to put here in the nucleus, and I'm going to put here of a dividing cell. All right, so that's when we call it chromosomes. All right, so each chromosome is composed of a single DNA molecule associated with proteins. Those proteins are those histone proteins that we just talked about. So before the cell divides, you have 23 pieces of DNA wrapped around these proteins called chromatin, and they're loosely packed. When that cell gets ready to divide, all of those pieces of DNA wrapped around those proteins condense and form these short, stocky structures called chromosomes. So that means that there are 23 chromosomes in humans because there's 23 pieces of DNA. So before they, condense, the DNA and the proteins of chromosomes are called chromatin. All right, and so the chromatin is found in the non-dividing cell. It condenses, the chromatin condenses to form discrete chromosomes as the cell prepares to, and that is to divide. So that's in the dividing cell. The nucleolus, we already talked about, is located within the nucleus and is the site of, what did rRNA stand for? Ribosomal RNA. Synthesis, the making of it. And so that is the nucleus. So attached to the nucleus, sometimes you have ribosomes. So going over here, coming back up to this picture, let's pick a darker color pink here. Here's your ribosomes. These are your ribosomes right here. They can be attached to the nucleus, and they're also attached to one of the endoplasmic reticulums. So let's look at ribosomes. This is where we said proteins are made, right? So they're the complexes, and they're made up of ribosomal RNA, or rRNA. I'll just write rRNA since we wrote ribosomal RNA just a minute ago. And those proteins, and where that they're made, remember where they're made? Made in the nucleolus. Ribosomes carry out protein synthesis in two places, in the cytosol on free ribosomes, meaning the ribosomes are floating around in the cytoplasm or cytosol all by themselves, or they can be on the outside of the endoplasmic reticulum. You know what? Let's write rough endoplasmic reticulum. Or they can be on the nuclear envelope. I just showed you the nuclear envelope. That's another place it can be. These guys are called bound ribosomes because they're stuck on something. They're bound to something. And so here is the transmission electron microscope of the rough ER. And so all these little dots on here are ribosomes. And what this is you're looking at a cross section. This is the cross section of the ER. And so there are ribosomes bound to the ER. Put in green, ribosomes bound to the ER, so they're actually stuck to the membrane of the ER. And there are free ribosomes floating around in the cytoplasm. So those are your two different kinds of ribosomes. No matter what type of ribosome we're talking about, they are made from a large and a small subunit that come together. So they look like little dots, but they're actually, there's a little bit more to it. And so they're made out of that rRNA and protein, small and large subunit. All right, so that's the nucleus and the ribosomes. Next, we're going to look at some organelles and how they all work together. And so concept 6.4 says the endomembrane system regulates protein traffic and performs metabolic functions in the cell. So we're going to learn about this endomembrane system. Endo means inside. So membranes inside the cell, and it's a system because they're all working together. So these are the organelles that are part of it. So the nuclear envelope, so we've already talked about that. The ER, the rough endoplasmic reticulum, Golgi apparatus, lysosomes, vacuoles, and plasma membranes. All right, and so these guys are either continuous, meaning that they're hooked together, or they're connected indirectly by vesicles that go from one to the other. So let's give the look at the ER. We've already talked about the nuclear envelope. The nuclear envelope in the ER the rough ER are connected. And so over here, back on this picture here with the nucleus is actually connected to the rough ER. And so let me show you this picture right here. This is the rough ER. This in darker purple is the nuclear envelope, two membranes. And you can see here that right here, the membrane from the nucleus goes and there's a tunnel to the membrane of the rough ER. And so therefore they are connected together that way. And so that's what it means by continuous. So the endoplasmic reticulum, we call it the ER for short, accounts for more than half of the total membrane in many eukaryotic cells. What does that tell us? If it's more than half of the total membrane, that there's a whole lot of rough ER or endoplasmic reticulum inside of a cell. And so a lot of that membrane. The ER membrane is continuous with, I just went over that, the nuclear envelope. There are two distinct regions of the endoplasmic reticulum, what's called the smooth ER. Why it's smooth is because the outside appears to be smooth, not bumpy, and that's because it lacks ribosomes. This is the type of ER that has no ribosomes on it. And then the rough ER, whose surface is studded with ribosomes. So the picture you have here in the next page is the picture I just showed you in color here. Let me put that up here again just so that you can see. I wanted to show you, I didn't point out the smooth ER. So here's your rough ER. You can see the little dots on here are ribosomes. And then connected to the rough ER is the smooth ER. So the rough ER looks like kind of like flattened sacs, all right, and it's a cross section here so you can see the inside of it, whereas the smooth ER are skinnier, kind of like tube-like. But they're all connected, but the rough and the smooth ER have very different functions, even though they're continuous and connected. So that's what we're going to look at next. What are the different functions of smooth and rough ER? So the smooth ER synthesizes lipids. So it makes lipids. It metabolizes and can break down. That means break down carbohydrates. It detoxifies drugs and poisons. So breaks them down so they become not toxic. And it also, they're specialized ER that store calcium ions. If you've taken an anatomy, you may have learned when muscles contract. Calcium ions are really important in that, and they're stored in a specialized ER called the sarcoplasmic reticulum. And so, it releases calcium ions so muscles can contract. So that's a specialized form of the smooth ER. So one point I want to make here with the smooth ER and about organelles in general is that not all organelles are in equal amounts in every cell. So there are tons of different types of cells. So, your muscle cell I just talked about is different than your cells of your eye and so on and so forth. So therefore the amount of smooth ER or any organelle can vary based upon the function of that cell. So the type of cell that has a lot of smooth ER is the liver cell. The liver cell has a lot of these because these cells do a lot of carbohydrate metabolism and is the site of alcohol detoxification. So what happens is that the liver filters the alcohol, let's say, out of your blood and breaks it down into non-toxic molecules. And then your kidney will filter out those molecules out of your blood. And so what happens is that your cells can actually adapt to that. So liver cells have a lot of these, and what can happen is that the more alcohol, so if somebody is an alcoholic, the more alcohol that they drink, the more smooth ER that cell makes, and so therefore it allows them to detoxify or get rid of that alcohol out of their bloodstream a lot quicker. So that's why people, if you've ever heard that if somebody is an alcoholic or begins to drink a lot, that they have to actually start to drink more alcohol to get the same effects that they used to get drinking lesser amounts, and that's because their liver is adapting to that, making more smooth ER. But what happens is eventually you can go through cirrhosis of the liver where it just puts too much strain on the liver. There's so much to do that it can actually kill the liver cells, and you cannot live without your liver. So that is not a good thing. So that's your smooth ER. The rough ER has these bound ribosomes, which means that then they probably have something to do with protein synthesis, because ribosomes have to do with making proteins, all right, which secrete glycoproteins, proteins covalently bonded to. Whenever you see glyco in front of something, it refers to carbohydrates. And then they distribute what are called transport vessels, and these secretory proteins, this means they will be secreted from the cell. What does secreted from the cell mean? They leave the cell. So the transport vesicles, you have these contain these secretory proteins surrounded by membranes, and it also helps to make membrane for the cell. And that'll make more sense here as we go along and we do some drawings here. So it's a site of protein synthesis. Let's look at, I'm going to go over here and draw a cross section. So of your rough ER. So this is your rough ER, and let's say rough ER here, and let's draw a ribosome attached to it. So this is the ribosome. I'm drawing it kind of big. It wouldn't be that big compared to the rough ER. So what happens is at the ribosome, what happens is protein synthesis, right? So we start to make proteins. So it's going to start adding one amino acid to another. So therefore you get this growing chain, a growing polypeptide chain. So it's hooking one amino acid to another amino acid to another amino acid and to another amino acid. And so this polypeptide chain is being made. And so eventually the whole thing, here's your polypeptide chain, comes over here, and what happens is the rough ER, so I'm going to draw another picture down here to kind of show what occurs. So there's your polypeptide made. Here's that same rough ER, same ribosome, and that finished polypeptide, what happens to the rough ER is the membrane starts bubbling out like this. All right, so it starts to bubble out, and so realistically, let me redraw the picture here. I'm not the best artist, so bear with me here, but well, I'll do it here. So this I'll just use this picture here. So the polypeptide, remember, so this membrane should be bubbling out like this. All right. So it's a little bubble out there. Pretend like this part's not here, and the polypeptide comes in. And then what happens is, you can imagine this pinches, and what you end up with is a little vesicle with your polypeptide in it. So here's your rough ER, still with the ribosome attached, and so on. So the rough ER has made the ribosome on the rough ER has made a polypeptide. The polypeptide bubbles out, and so this right here is called a transport vesicle. And so it's a little vesicle that can transport this polypeptide, all right, from one place to another. So it was made here in the rough ER. All right, so we just said that the rough ER is going to make these secretory proteins. So this is the beginning of a protein, a polypeptide chain that's going to be secreted from the cell, but it's not yet secreted from the cell. And it also helps to make, is a membrane factory for the cell, so helps to make cell membranes. And so that's where the next thing comes into play, the Golgi apparatus. So that's where actually the transport vesicle that buds off of the rough ER goes. The Golgi apparatus consists of flattened membranous sacs called cisternae. And so the functions, what are the functions of the Golgi apparatus? They modifies products of the ER. So like, example, the proteins that we just talked about. So this protein or this polypeptide is going to go to the ER and it's going to get modified. So that, what does that mean? It chemically changes it a little bit, adds some chemical groups and so on. So what that's going to do is change how the R groups in that protein bond and therefore help to make the right shape. So it modifies it, and afterwards the protein will be in the right shape. It manufactures certain macromolecules, sorts and packages then materials back into transport vesicles. So after they're done with that. And production of cell membrane as well. So this is like the transport vesicle, like the rough ER. So let's draw that here. Well, first of all, let's look at this picture here. So this is what the Golgi apparatus looks like. So these flattened sacs. And so what's going to happen is it has a receiving side and a shipping side. You don't need to know cis and tran, the names there. But basically the transport vesicle brings its contents to the Golgi apparatus. It goes through, the contents go through the Golgi apparatus. It's going to modify it, and then it bubbles off in another transport vesicle. So let me explain that in my little picture down here. So I'm going to make the Golgi apparatus. So what color should I make it? I'm going to make it light blue. So here's your Golgi apparatus. All right, so there's these sacs. All right, and there's all these different sacs here, the Golgi apparatus. So this is the Golgi apparatus. I'm just going to put GA. So what happens is that transport vesicle comes over here from the ER, so from ER, rough ER, and binds to, fuses with the membrane of the Golgi apparatus. So fuses with that, and its contents go in. So the polypeptide goes in. The polypeptide, these guys are connected via tunnels and so on. So the polypeptide moves through, and so this green here is, it's modifying it, adding chemical groups and so on, modifying the protein. And then eventually, what buds off here, the membrane of the Golgi apparatus will grow out and bud off like this. And another little transport vesicle. And your finished protein will be in there. It'll pinch off here and here. And you end up with another transport vesicle with your finished polypeptide. I'm going to make it curved here that shows it's a three-dimensional shape. So this is your transport vesicle. From the Golgi apparatus, and I'm going to put here with the finished protein. All right, and then remember that back over here, the polypeptide that's made, the protein, going back to the rough ER, it says makes secretory proteins. So what does that mean? It's going to be secreted from the cell. So guess where this transport vesicle goes? This transport vesicle goes to, this is all happening inside the cell. So this is the cytoplasm in the cell. Goes to the cell membrane. So right over here, this right here, we're going to call it the cell membrane or plasma membrane. Remember that lipid bilayer? And so what happens is this transport vesicle comes over here, fuses, so this is going to fuse and become part of the membrane of the cell, and therefore secretes the protein. So the protein leaves the cell. And so then this part right here, so that's the secretory protein, but remember, it said here membrane factory for the cell and a production of cell membrane. So this fuses with this, and this actually increases the amount of cell membrane because you're adding membrane to it. So these are all part of this endomembrane system because even though like the Golgi apparatus membrane is not connected to the rough ER, it's the same type of membrane and they can fuse together and are kind of interchanged. And so therefore things can move via these transport vesicles from one organelle to another. And so this is the endomembrane system, and that's how we get our secretory protein. And for the last one I want to talk about in this video is with lysosomes. Lysosomes are a membranous sac of enzymes. So it's just this little sac, and it has enzymes in it that digest macromolecules. So what does that mean? Carbohydrates, lipids, proteins, and so on. So the enzymes are enclosed in here so they don't digest just anything. So they digest only certain things that are put into the lysosome. So these enzymes work best in acidic environments, and so inside the lysosome is acidic in nature. So the enzymes in the lysosomal membranes are made by the rough ER. So this relates, I'm going to put ER, I'm assuming now you know that ER stands for endoplasmic reticulum, and then transferred to the Golgi apparatus for further processing. So what does that mean? Let's go back to this picture here. So I'm going to use another picture here, is that sometimes the polypeptide that's made here buds off of the rough ER into a transport vesicle, goes to the Golgi, and is actually then becomes a lysosome. So that's another pathway, lysosome, all right, that has digestive enzymes, and the enzymes are the protein that was made in the rough ER. So the enzyme, I'm going to put the same protein there, is what's actually made. So what we have here is one thing that can happen through the endomembrane system is the transport vesicle. All right, and then we get a secreted protein. The second thing is that it actually doesn't become a transport vesicle. It actually becomes an organelle of the cell, of animal cells, called the lysosome. All right, and so this actually stays in the cell and has a job. So let's look at what that job is. So some types of cells can engulf another cell by what we call phagocytosis. This forms a food vacuole. So what does this mean? It means that this cell, we're going to look at this picture here. Here's the membrane of the cell. What it does is the cell membrane buds in. So this is let's say, a unicellular cell, cellular organism. This is how they eat. They don't have a mouth. They're only one cell big. So therefore, how do they get their food? And so this is their food right here. All right, so this is food. And they get their food by indenting their membrane, taking it in. This membrane pinches off, and you end up with a food vacuole. So that's what this was all about, that what we just wrote. We have a food vacuole, and inside the food vacuole is the food. So now the organism needs to digest the food. Just like when we eat, our digestive system breaks down the food for us into small enough molecules that our cells can use. In unicellular organisms, they don't have a digestive system. Again, they're only one cell big. So in comes the lysosome. So here's your lysosome with the digestive enzymes. Notice the lysosome fuses with the food vacuole. So let's do, the food was green. And let's do the enzymes, I don't know, let's do the enzymes red. So the enzymes in here were red. So the enzymes start mixing with the food and you get digestion happening. So that is one job of the lysosome, is to digest food for those unicellular organisms. So what happens here, a lysosome fuses with the food vacuole, like we just looked at, and digests the molecules inside. That's the first thing. The second thing, they also use enzymes to recycle their cells' own organelles and macromolecules called autophagy. So sometimes organelles get old. So you might have an old mitochondria or something like that. So here you have it engulfing the lysosome, fusing again with them and digesting the mitochondria. And so mitochondria happen to divide, and so we constantly make new mitochondria, but then they age, and we want to get rid of these old organelles. And so the lysosome fuses with them. Those enzymes in the lysosome digest it, and we get rid of our own organelles. And then they're also involved, we'll talk about this several times during this class, involved in cells, programmed cell death. That's called apoptosis. So sometimes we want the cell to die. And so oftentimes the lysosome, those digestive enzymes in the lysosome will actually kill the cell. And so an example of a time when you would want that to happen is sometimes during development. The cells of the tail of a tadpole, which are digested as a tadpole changes into a frog, and so a frog doesn't have a tail, and so it destroyed those particular cells. An example of a bad example of the lysosome gone awry, absence of a particular lysosomal hydrolytic enzyme. Hydrolytic comes from hydrolysis, which remember is to add water, which means to break down molecules. So this enzyme is going to break this down, and but there's an absence of that. So then this is Tay-Sachs disease, in which an enzyme is used to digest lipids, and it's absent in those people, and so they get accumulation of lipids in the brain, which causes a genetic disorder. And so that is your lysosomes. And so the lysosomes are also a part of the endomembrane system. And this is where I am going to stop for this video.